in the elongation of the cell, the cylindrical or the lateral growth of the cell during when the cell is not dividing. When the cell simply elongates in length, it simply grows. And elongosome is only present in rod-shaped bacteria, right? Because the spherical bacteria are not becoming elongated. It is only present in non-spherical or rod-shaped bacteria, whereas the divisome complex is present in all shapes of bacteria, and it's highly preserved. In fact, the divisome complex is also present in mitochondria, because we talked about how mitochondria are very much similar to bacteria. So the divisome complex from mitochondria is called a mitochondrial divisome complex. And the divisome the complex is only assembled when the cell is actively undergoing cell division or binary fission. And the divisome complex is always present only in the middle of the cell. And it has a unique set of proteins, the Fitz proteins. So the Fitz protein help in the constriction of the inner membrane and the outer membrane. And the peptidoglycan precursors are added to the center, the Z ring or the septum right in the middle. Whereas the elongosome is present throughout the length of the bacterium, and it adds peptidoglycan throughout. So it just goes and spirals, just like in the image, the very first one here, it goes throughout the cell. It just moves all along the length of the cell, not just in the middle. Okay, does that make sense? And it's got unique proteins in it. It's almost similar. The two complexes are similar in the sense some proteins are similar, like you can see the MUR, MUR-B protein, MUR-C, MUR-D. Those are all the actin or the actin-like proteins. So they basically help in the movement of the complex. Whereas the Fitz proteins, there's the Fitz-Z, Fitz-A proteins, they help in the constriction of the inner membrane, the outer membrane, and they're present only in divisome. They're not present in elongosome. And then the penicillin binding proteins are the ones that actively recruit peptidoglycan molecules into the growing peptidoglycan polymer, either at the divisome or at the elongosome. Okay, is that clear now? Any questions? Okay. All right, so we talked about different ways to measure cell growth. So we talked about viable cell counts. We talked about direct method and indirect methods. Direct methods using hemocytometer, using microscopy, and then indirect methods using serial dilution and pour plate methods, spread plate method. So there's no confusion in what we've learned about measuring cell growth, right? So we're going to talk about a new method. It's called the most probable number. So just like the title somewhat suggests, the most probable number is a statistical model that gives you a rough estimate of how many viable cell counts are present in a sample. And this technique is mostly used for samples like you may have a water sample or a sewage sample. In microbiology, we should be able to, produce that estimate. If you have a sample of some sort, you should be able to test how many bacteria are present, or a specialized technique is like a commonly used technique is how to count number of coliforms present in a sample. So coliforms are all the genus Escherichia, for example. There's a gut, it's a gut, we know about E. coli. It's a gut bacterium, which is often used as a fecal indicator of fecal contamination. So say you have a water resource, potentially it's a drinking water source, and you want to test if the water has fecal contamination. Then you want to take a sample of that water, and then you want to run this test. And by the end, you'll be able to tell whether the water is contaminated with coliforms, and if yes, what is the concentration? Normally there's a limit. If you have these many cells per liter of water, then it's still considered safe. And then if it's higher than a certain number, then it's not safe for consumption. Because even in human body, for the infection to happen, we need, this is called the infection load, or can't remember the term, but if the number is greater than a certain amount, then that means this likely chance of causing infection. And if the number is fairly low, our body can clear it off. So, so this test will give you that number. So the way it works is you take, so you have the sample water, and then you take 10-fold dilution, like different amounts. So you take 10 mL or 1 mL and 0.1 mL, for example, and then you inoculate that. So there is 3 steps to this. So there's a presumptive test, confirmatory test, and completed test. And you perform each of those steps using different media. So by the end of the completed test, you will know how many coliforms are present and in what number. So in the first step, the presumptive test, so you take different volumes, so 10 ml, 1 ml, and 0.1 ml, and then inoculate that in lactose broth or McConkey broth. And if you have growth, so presumably you will have more positive or a positive test in tubes where you inoculated 10 ml, right? Because a higher volume, there's a likely chances of your coliforms or the bacteria you're testing is present. And then 1 ml has a less likely chance, and 0.1 ml has even more less. And then if you have a positive test, let's say your tubes are showing positive results, then you get a number. So either in the first 10 ml, you get 5 positive, all of them are positive. And then in your 1 ml, there's only 2, and in the 0.1 ml, there's only 0. So take those numbers, 5, 2, and 0. You take that number and input that into a probability table. So in the textbook, there is a table. Using those numbers, it gives you a rough estimate of per liter of sample, there are these many bacteria. And then you move on to the next step, which gives you what are the chances, like an even higher probability that the coliforms are present. And then you do that using lactose broth or Brilliant Green lactose fermentation tube. So here, so coliforms are lactose fermenting, meaning that the pH of the medium changes. So the pH becomes acidic. So there is an indicator, a dye indicator, that usually changes the color from, I don't remember exactly, I think yellow to pink, I think. So the dye basically changes its color when the media becomes acidic. And then you also, so coliforms also produce gas. When they're doing lactose fermentation, they produce carbon dioxide. And then you test that by putting a small tube inside your test tube, and that's called a Durham tube. And if there is accumulation of gas, if there's air bubble inside, it means it's a lactose fermenting, gas producing coliform is present. And then you move on to the third completed test, where you take a little bit of that organism and you streak it on a plate. And if it's a coliform, you'll get a metallic sheen. So just like how in the picture, it's metabolizing the, so how does the color change? I don't really know the biochemistry of it, but if it's a coliform bacteria, then you get a green metallic sheen colonies on your plate, which is a confirmatory test that is a coliform organism, and you get the number of those cells present from your very first step. And then the second and the third one is just to confirm that it is indeed the coliform or the E. coli that's the one that's responsible for making the color change. And yeah, and then the number you get from the probability table. And there's a link at the bottom that I put. There's a really nice video, very clear, that goes through the different steps of the MPN test. The video was about 11 minutes long, and I didn't want to show that here in the class because it would take a big chunk of our today's lecture. But I highly recommend you go through that video. It's really easy to follow. And there's also a person performing the test. So it's just really nice visual for you, because likely you won't be doing that in the lab. So it's a nice visual for you because the person go through the different different steps and then explains what media is being used, inoculation, how to sterilize your lube, and everything. So it's a really nice video. I recommend you watch it. Is MPN clear? Any questions? pH and bubbling, both. First one is just growth, as far as I understand, but yeah. But the lactose broth, it also has lactose in it. So, but the McConkey broth might be showing a color change. I don't know 100%, but I think the first one is just growth. The second one is pH change. It really depends on what media you're using. If you're using the media from the confirmatory test, it will still show you a color change. Yeah, it's just the lactic acid just showing the color change. And then, yeah, the material that I've referred to, it's done in the second test. The confirmatory test is where it shows the color change and the gas production. Yeah. Yes. Yeah, the color changed from pink to yellow. Yeah, not the other way around. Anyone else? No questions? Okay, so another method to test bacterial growth is called optical density, or simply called absorbance. Absorbance, fairly straightforward, but very powerful and very routinely used method to test bacterial growth. Have you used a spectrophotometer before? Because it is so routine, some of you might have, you guys have already done it. Okay, well then maybe I'll just quickly keep it brief, but there might be other people on Zoom call that may not have used it. So it essentially measures the turbidity. So as you know, bacterial media, after you make the media, you sterilize it, looks clear. And then you inoculate it, leave it in the incubator for a day or two, or maybe longer, depending on the type of media, you will see a turbidity. And the spectrophotometer is essentially measuring the turbidity. So it's measuring how much of the light is being absorbed, and the remaining light is obviously transmitted, and that ratio of the light transmitted to how much the light is absorbed is represented by a number. And that number represents the concentration of the cells in your media. Now, how many bacteria are there? It doesn't say how many bacteria, it just gives a rough estimate of what stage of the culture is in. If it's mid-log phase, you normally get about 0.4 to 0.5 OD. And then if it's late stationary phase, it's usually greater than 1, 1.2, 1.5. If it's early log phase, it's close to 0.1 or 0.2. So very routinely used. It also measures the dead cells, because obviously dead cells also scatter light, but then it's not as big of an issue in microbiology because we rarely use culture that's past stationary phase, unless somebody's doing research specifically on that area. In that case, they just come up with a different way of measuring cell growth. But in microbiology, we're almost always interested in the log phase or the early log phase, when the cells are super actively dividing. That's when we're most interested in them, in which case the dead cells are much fewer in number and then doesn't really contribute much to the variation in the OD. And optical density is always measured at 600 nanometer because it's safe for the bacteria, because I think some time ago we were using UV spectrophotometers, but then UV is not safe for bacteria and causes mutation. So now 600 nanometer is routinely used. I think that's the wavelength where the bacteria, it's appropriate because that's when they show the light scattering and easy to measure and safe for the bacteria. And OD is always represented by just a whole number. It's a unitless because it's a ratio of transmitted light to absorbed light. Yeah, fairly straightforward. Normally you have a cuvette with the two sides that are transparent that allow the light to pass through. And then you put a 1 ml of culture in it, stick it in the spectrophotometer, and then it gives you a number, which should be a rough estimate of the cell growth or the stage in the life cycle, the cells. Okay, so we're switching gears a little bit. So we've gone through the cell division and binary fission and all the, so the process of binary fission and then generation time, how to measure cell growth. That's mostly assuming the cells double in number or based on binary fission. But then now we'll move on to alternative modes of cell division. So as I mentioned, binary fission is the most common and the most well-studied. In fact, I don't like the word most commonly used by bacteria because we don't know of all the bacteria that are in nature yet. So we only know a certain portion of the bacteria in nature, and the ones that are easy to culture and that are easy to be studied, they undergo binary fission. But there are other ways that the bacteria undergo division, and that's by asymmetric division and budding. They kind of fall because budding is also asymmetric. And then there's fragmentation, which I'll go through after this. So when we talk about budding, example, always the most well-studied is Saccharomyces cerevisiae, or the common yeast. So in budding, I'll talk about asymmetric division after I talk about budding. Yeah, you probably know there's usually the parent cell, which is, in case of yeast, it's circular. And then when the cell is ready to divide, so the nucleus gets divided, and then on one, at one point along the cell wall just becomes softened. And then the nucleus and the part of the cytoplasm just essentially moves towards the soft spot, and then which eventually turns into a bud, just like in the picture. And then the bud grows in size and then eventually separates from the mother cell. And there's usually a scar that's left behind, which is often easily viewed with, not with the light microscope, but with the AFM. Definitely you can see a scar. So that's budding. And then the bud obviously turns into a new daughter cell, and then that undergoes, budding is just a very commonly used asexual form of reproduction by Saccharomyces cerevisiae. And then it's not just yeast that undergoes budding. There are a few bacteria that also undergo budding. It's really interesting, actually. So I have a couple of examples here: Hyphomicrobium and Caulobacter. So those are the commonly fairly well-studied examples. These two bacteria, they almost give me the impression that it's that kind of in between a bacteria and a fungus, but it is a bacteria because they're prokaryotic. So in case of Hyphomicrobium, the image that I have here, what the bacteria does when it's ready to divide is produces an elongated filament from one pole of the cell. And that elongated filament is called a prostheca. And the nucleoid undergoes division, and the newly divided nucleoid basically just moves through the filament and then into the other end of the filament, which becomes like a bulb almost. And the nucleoid goes inside that newly formed protrusion, which eventually separates into a new daughter cell. And another example is Caulobacter, just similar, except Caulobacter, it attaches itself to a substratum. And then, so I have the life cycle here in the picture here at the bottom. So the free living cell has a pili and a flagella. It's a swarmer or it's a swimmer. And then when it's ready to divide, it just goes and attaches itself to a surface, but using a protrusion will hold fast. And then there is a stalk at the bottom using which it attaches itself to the surface. And then the other pole elongates and then forms into a daughter cell once the nucleus is divided. And that the daughter cell then eventually separates. So the division always happens at one pole. It's usually the one pole that elongates and then turns into a daughter cell, and that becomes free living and then just swims away. And then that eventually attaches itself to another place, and then the division goes on. So the hyphomicrobium that I talked about earlier, it's interesting because if you look at hyphomicrobium under a microscope, it looks like a mycelia. Do you guys know what mycelia means? So the mycelia is a term normally used for fungi because they form this filamentous mass of hyphae. It's another technical term. If you don't understand, it just means long hair-like structure which has septum, and it's nucleated. So each septum is a cell, because fungi are multicellular. So they just form long filaments of hyphae, and then they produce aerial hyphae, which has conidia and produces and that's what is, those are called mycelia. So hyphomicrobium is actually a bacteria, it's prokaryotic. But then it forms these long filaments that are somewhat similar to mycelia, except they're not separated, or it's just, the whole thing is just one cell. And then a new cell is formed at the end of that filament and then turns into a daughter cell. Well, based on the morphology, it just gives me this impression that it might be like an, in between evolution between bacteria and fungus. And in the next slide, we'll talk about actinomycetes, which is also similar, but I don't want to bring it up right now. I don't want to confuse you. And any questions so far? I'll talk about one more thing, and then we'll move on to fragmentation. Yeah, Caulobacter. To me, that looks a lot like binary fission, at least for the but yeah. Could we differentiate that one, or why is it? Yeah, no, that's a good question. Yeah, and the question, I'm just going to repeat the question to people on Zoom. So Thomas was just asking why Caulobacter looks, the division, if you look at the image there, it looks a lot like binary fission. You're right in the sense that the daughter cell has the same size as the parent cell looks like. And that's why you think it should be similar to binary fission. Yes, I mean, yes, but I guess the main difference is in binary fission, the division is not at the pole, right? The elongation does not happen at one pole. It happens right at the middle of the parent cell. But here it's just one pole that's elongating. That's the main difference. One side of that bacterium, like that long one. Yeah. It's just a one, yeah, yeah, exactly. Yeah, it's just a one pole that becomes long, and then that separates and goes away. So it's not, the division is not happening right in the middle. Yeah. Yeah, no problem. Any other question? Before I talk about this really interesting case of budding. There is one bacteria example that we know of called Epulopiscium species, and then Toxoplasma gondii. So Toxoplasma is a protozoan. It causes toxoplasmosis. You might have heard about like why pregnant women are not advised to clean up cat litter, because Toxoplasma is one infectious organism that can cause serious birth defects in pregnant women. So it's a protozoan, and the budding process in Toxoplasma is internal budding. It's not external, just like how it's in Saccharomyces. So in case of yeast, the bud is formed on the outside of the parent cell, and the bud separates and turns into a daughter cell. But in case of Toxoplasma, it's internal. So what happens is the nucleus gets divided into two daughter cells internally inside the main parent cell. And it is assumed that the daughter cells basically consume the parent cell, and the parent cell basically bursts open and the daughter cells are released, and they basically swim away. So the parent cell basically dies in this case. The parent cell gets consumed, and then the buds are formed inside the parent cell. And it's similar in Epulopiscium species. So it's a big bacteria, in the sense the size is huge, and it lives inside the gut of surgeonfish, tropical surgeonfish. And what happens here is also similar. The daughter cells are formed inside the parent cell. And it happens, the life cycle of this bacteria coordinates with the feeding habits of the fish. It's really interesting. If you want to take a look, there's a really simple Wikipedia page, and there's might be other papers. So in the morning, there's just the daughter cell, the small daughter cell, but as the fish feeds during the day, these daughter cells increase in size, and the nucleoid would have divided. There's 2 cells present inside the parent cell. And then by the end of the day, these daughter cells burst open from the parent cell, and then they, again, go through the same cycle the next day. So that's another really interesting form of budding, but quite different than what the Saccharomyces shows. Any questions? Okay, let's talk about fragmentation. So some cyanobacteria and actinomycetes show fragmentation. So here what happens is the nucleoid gets divided more than once inside the parent cell. And there's simply accumulation of the wall material around each of those nucleoids. And then the parent cell becomes fragmented, and then each of those parts basically float away and then turns into adult organisms. Cyanobacteria, they sometimes are filamentous, even though they're prokaryotic and they're unicellular. So they form filamentous, long filamentous, sometimes they have septa. And what happens is each of those filaments basically get broken off, and then they float away and then turn into other, they start dividing, and the life cycle continues. And it's similar to actinomycetes too. So actinomycetes, until recently, they were considered to be fungi, not bacteria, because their morphology is so similar to fungi, in the sense that, as I explained, they have mycelia, they have hyphae, they even produce spores, like aerial spores, all similar to fungi, except they are prokaryotic and they're bacteria. I mean, recently it was, I guess not so recently, they found out they're actually bacteria. So these mycelia, again, once a nucleoid undergoes division, they get broken off, and then they get attached somewhere else, and they turn into, okay, I guess, yeah, the second picture is actually the life cycle of actinomycetes. So one way they reproduce is by producing free spores. And the other way is this mycelia, which gets fragmented, and then they attach to other parts, and then, or other surfaces, and then they turn into a mature organism. So yeah, actinomycetes, they're really interesting again. So when the first rain hits, like in tropical environments, it's very easy. Even here, when it rains, that marshy, the muddy smell you get, it's because of actinomycetes. So they're present everywhere. In fact, they're also present in humans. They don't cause any disease, but they're just simply present as the normal microflora, although I think they're symbiotic. And they also form symbiotic relationship with the plants. They help with the nitrogen fixation. They have a really important role in nature. That are like fungus, but except they're bacteria, and then they undergo fragmentation. Fairly straightforward. Any other questions? Any questions related to this? Yeah.
And so when that breaks apart, individual cells that are then divided into and are they binary fission after they occur?
They both ways. They can undergo binary fission or fragmentation.
Okay.
Yeah.
So when it's breaking off, it's not really reproducing and breaking off. It's breaking apart and then they start at the column.
Yes.
So the underlying mechanism of reproduction would just be the binary fission, right?
You know what? To be honest, I don't know if cyanobacteria undergo binary fission. I don't know that 100%.
Some kind of division, but the fragmentation itself is just distributing itself, right?
Yeah, but there's also, I mean, it is a reproduction in the sense that each of the filaments, the nuclear still has to divide, and then they become long. At some point in time, they get broken off, they get fragmented, and then becomes a new organism. It is a reproduction, not just fragmentation.
I guess if you think of the filament itself as breaking into two filaments, I was just thinking at the layer of the cell itself.
Yeah.
Because each filament is made of a number of things, right?
Yes.
Okay.
Yeah. 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 Yeah, I mean, at this level, I just even question whether they're unicellular because there's a lot of cells attached to each other. But each cell, I guess, is an organism by itself. So it's not, yeah. But anyway, I guess it gets, sometimes you cannot differentiate bacteria from fungi. They exhibit features that are mixed, and nature that sometimes doesn't fit in boxes of this or that. Anything else? Okay, so let's talk about biofilms. I kind of talked about biofilms in the past, just mentioned it a few times probably because they're just, again, really interesting feature exhibited by bacteria and by fungi, by algae, all kinds of microbes. So biofilms are complex communities of dynamic ecosystems. So in nature, there are biofilms everywhere, pretty much. If you haven't paid attention, you go to a lake or any sort of wet environments, and rocks are usually covered with like a slimy layer. Those are biofilms, and also the green floating, what we call algal blooms. The floating green mess sometimes you see in lakes or any kind of water resources is usually biofilm. So they're pretty much everywhere. In fact, the water bottle you might be drinking, if you didn't clean it for a few days, there's usually a layer of biofilm. Nothing to be grossed about. They're often probably not unsafe, but there are biofilms that are unsafe, which I'll talk about in a little bit. But yes, water treatment plants and any sort of piping that, as long as there is some kind of organic molecules dissolved in that water, bacteria find a way to use it, get attached to that surface, and then once it's attached, it builds up into, becomes thick and thick, then just gets detached by the flow of the water, goes and gets itself attached somewhere else, and just keep multiplying and keep growing and keep becoming complex. And they could be just single type of bacteria. It could be a mixed bacteria and fungus. Sometimes it's mixed with algae. The really complex type of biofilms exist in the environment. So this is an image of an SEM, like a very low resolution SEM image of E. coli biofilm that I had taken. And then in the next slide, I'll show you how they form. And then this is a more zoom in picture image of the E. coli biofilm. So normally biofilms start with one cell, the floating one swimming cell that is producing, we talked about different layers of layers present in the bacterial cell envelope, right? We talked about membranes, there's a cell wall, then some bacteria produce capsule, and then slime layer. So it's the slime layer that plays an important role in the formation of the biofilm, because it's made of extracellular polysaccharide, essentially a sugar polymer that helps in the attachment, and also pili. pili also helps with the formation of the biofilm along with the slime layer. So the bacteria swimming and then attaches itself to a surface, it divides and produces more slime, and then the biofilm essentially becomes thick that way. And then it recruits other organisms, could be even a fungus, or could be other species of bacteria. And then the biofilm then becomes more complex and a very organized structure. So if you look at the zoom in image of a SEM of a biofilm, there is bacteria, obviously, and there is the more important component is the extracellular matrix, which is so often 50 to 90% of the total dry mass of a biofilm is just the extracellular matrix. It is made up of mostly sugar polymers, but then it also contains waste material like proteins, nucleic acids, contains cell debris. Sometimes it also has cellulose because there's bacteria that produce cellulose, often from even a thicker matter of biofilm. And then there's channels inside for the movement of water, movement of gas. It's a really complex system these bacteria produce. And they're highly resistant to any kind of disinfecting material that you might want to use to get rid of it. They have antibiotic resistance. There's lots happening in these biofilms. There's exchange of genetic material. So competence, I kind of mentioned it a little bit, where the bacteria take up the DNA from other organisms. Biofilms are notorious structures where there is transformation, or transformation happens by the process of competence. Don't get confused with different terms, but they're basically the same. If the bacteria is competent, then it undergoes transformation. So transformation is a method where pili attaches itself to a piece of free-floating DNA and then takes up inside the cell, which gives its additional feature. Kind of talked about that. And biofilms are perfect environments for bacteria to undergo transformation. And that's why they're a really big issue in the clinical environment because that's how bacteria acquire antimicrobial resistance. In hospital settings, in all the clinical tools that we use, and then there is obviously in the hospitals, there's tons of antibiotics used. One bacterium acquires antibiotic resistance, and then it just gives it to another bacterium. And then that's how bacteria become antibiotic resistant. So this is kind of figure that I had made for one of the book chapters. Kind of talks about how like a multi-species biofilm is formed. So active motile bacteria can become static and attach itself to a solid surface using surface adhesions or pili or slime layers. And then once it forms like a fully mature biofilm, the loose cells can escape the biofilm, or sometimes just by the sheer forces of the water floating through in the environment, parts of that can get dislodged and then form new communities. And then there's transport of nutrients, gases, biomolecules occur through the matrix, essentially what I just mentioned. It's got pores and channels, and then they show resistance to disinfecting agents and antimicrobial agents. And then, yeah, they could be made with algae, cocci, rod-shaped bacteria, sometimes even fungus, really complex communities. Yeah, I kind of went through it. First, once the bacteria gets attached to a surface, within a certain period of time, it's still reversible. So the bacteria can still detach itself and then still turns into swimming stage. So at that first stage, when the bacteria attaches to a surface, it's reversible. But after some time, once the biofilm becomes mature, it becomes irreversible at that point. It's really hard to get rid of. And then there's cell division over hours or days. Once the community is formed, it's really unpredictable on what the biofilm can become. Either it becomes complex, or if it's stays that can stay there for months or even longer than that. Yeah, again, I kind of mentioned how big of an issue biofilms are in human health. We do have good biofilms in our body. Like for example, in our intestine, There is biofilm of bacteria that are beneficial to us. They help us with vitamin production or digestion of our food, and maintenance of this healthy biofilm is really important. And on the other hand, we have biofilms that are not so good, like the dental plaque, for example, that causes cavities. And then there are some biofilms like those produced by Pseudomonas aeruginosa, which is an opportunistic bacterial pathogen. It's present normally in your body, but as soon as your immune system goes down, it attacks you. So biofilm produced by Pseudomonas around wound, like open wounds in hospitals, is a huge problem. They're really hard to get rid of, and then they're very easy to develop antimicrobial resistance because they often form biofilms, which are very hard to get rid of. People that struggle from cystic fibrosis often have biofilms produced by Pseudomonas aeruginosa. Again, very, very hard to treat and very hard to get rid of. Yeah, so biofilms, bacteria that are embedded in biofilms are very hard to get rid of. So some of the techniques that are currently being used are sonication. And again, you cannot use that on humans, but sonication is for like small materials, like small pipes, or sonication is using sound vibration to dislodge biofilms. And then you can also use enzymes that can break down polysaccharides, like amylases, for example, that break down sugar or the different components of the biofilm. And then when you combine enzymes with your disinfecting agents, that can be more effective. That's how in industries, all the pipes, so I kind of talked about the fermenter and how the continuous culture, all those pipings, biofilm is a huge issue. So often enzymes are used to clear up biofilms. And then, yeah, all the tools that are used in clinical settings, like contact lenses, catheters, even breast implants apparently have issues. So yeah, it's a huge issue in the clinical setting. Let's see what time it is. Okay, 5 minutes. Yeah, there's an article that I suggest you read, like a really simple article in Frontiers, which talks about how biofilms can make you sick and how to combat them, to talk about the techniques that are used in clinical settings to get rid of biofilms. Okay, so last thing for today. Any questions on biofilms? Yeah. Do I think it's a biofilm? Is that the question? Do I think gut microbiome is a biofilm? Yeah, I think so. Yeah. Why it's a biofilm? Well, it's a complex community, right? Yeah, anything that's, it's a film and it's a community of multiple organisms. It's a very, it's an ecosystem inside our gut. Bacteria are helping each other, there's mutualism, there's antagonism. Yeah, anything like a community of bacteria that are, yeah, I would call it a biofilm. Yeah. Do you have any arguments on why it should not be called a biofilm? Yeah. Definitely. Yeah, I know. I think I know where you're coming from. I mean, it's probably not forming like a static film as you would imagine on a plastic pipe or something. Like our gut is obviously different. Our body is monitoring how how proliferate or how advanced a biofilm gets, because our immune system is also monitoring it, so they don't grow beyond a certain level of complexity, right? I almost imagine. Yeah, I mean, I don't think there is a film in our gut, if that's what you're expecting. They're just a community of bacteria that are present in the wrinkles in your intestine. I don't know, use your imagination. But it's definitely a biofilm, but not the kind that you would see in outside, like on the surface of a rock in a lake or something like that. Yeah. Does that satisfy you? Any other question? Okay, let's talk about quorum sensing. This is again a really, really interesting phenomenon. So we think bacteria are really simple. They're just eating and dividing. But then the quorum sensing is a really Interesting technique. It's fairly new, still under research. Quite a few papers are coming out because it's, again, quorum sensing is very related to virulence, because that's how bacteria regulate not just virulence, but a number of different processes during the life cycle is controlled by quorum sensing. So quorum sensing regulates gene regulation. You understand the principles of gene regulation, do you? A little bit? You don't need to go in depth. So what it does is the bacteria, when their cell population increases in an environment, they produce certain chemicals. And those chemicals go and bind to the different parts of the gene, essentially the entire genetic material. So as you can imagine, some of them I have listed here: symbiosis, virulence, competence, conjugation, antibiotic production, motility, sporulation, biofilm formation. Every little phenomenon the bacteria shows is regulated by these compounds. And so, yeah, quorum sensing occurs through release of chemical molecules, and they're called autoinducers because the bacteria themselves are producing it. And then those compounds that are produced with bacteria have an influence on the entire population of bacteria in that system. It's really neat. So essentially, they're just monitoring like a community level response. And the autoinducers are produced by the individual cell of the community. So yeah, really interesting. The bacteria, the gram-negative bacteria produce acyl homocyanin lactones. So the structure, I've put a structure just for reference. And then gram-positive bacteria produce oligopeptides to communicate. So these oligopeptides are homocyanin lactones. They're produced in larger concentration when the bacterial population becomes more and more dense. And then they control the gene expression of all the different processes that I've listed here. What else did I want to tell you about that? Yeah, so that paper at the bottom is a really nice paper that talks about how quorum sensing plays a role in virulence, because they also do that inside the human bodies when they're causing an infection. And then they monitor the cell division and the growth cycle. One cell influences basically all the different, not one cell, but the cells essentially regulate the processes of the rest of the members of the population. Yeah, the paper has additional information, but yeah, quorum sensing is just a way that the cell communicate via producing chemical compounds. We have time. Oh, it's time, so that's perfect timing. So that was the last thing, and I just wanted to introduce the topic of quorum sensing. You don't have to go extremely in depth about that. But if there are no questions for today, I will see you on Friday.